The overall goal of this procedure is to monitor chimerism in the stem cell transplant setting.
This method can help address key questions in the transplant field, such as engraftment after hematopoietic stem cell transplant for patients with hemoglobinopathies. The main advantage of this method is that it combines flow cytometry-based cell sorting, colony formation assay, and subsequent analysis of short tandem repeats to assess the proliferation and differentiation of progenitors in the erythroid compartment.
The implication of this technique extends toward therapy of graft rejection because it may help identify patients susceptible to disease relapse and initiate subsequent immunomodulatory strategies. Furthermore, it also can be applied to future clinical studies, stem cell research, and design of gene therapy trials.
Visual demonstration of this method is critical as the processes are difficult to learn and need to be performed by trained personnel. Demonstrating this procedure will be Elisabeth Hoflehner, a technician in my lab.
To begin, generate a single-cell suspension of the bone marrow sample for FAC staining. To do this, place a cell strainer containing the bone marrow aspirate on top of a centrifuge tube. Use the syringe plunger to force pieces of tissue through the mesh. To collect all material, rinse with a few milliliters of suspension buffer. Add 3 milliliters of gradient medium to a centrifuge tube and carefully layer the single-cell suspension onto it.